So our next speaker is Wendy Huang from the Department of Cellular and Molecular Medicine at UCSD. Wendy studies transcriptional and post-transcriptional regulation of gene expression in the immune system during states of health and disease, and she's using cutting-edge technologies in the area of molecular biology, microbiology, proteomics, genomics, and genetics. So today we'll hear from her about regulating T helper cell immunity from serum amyloid to RNA helicase and its associated long non-coding RNA. Wendy.
Thank you, Steve. Can people hear me? Thank you, everyone, and thank you very much for the organizer for inviting me to share of our latest work. So actually, I just finished my postdoctoral fellowship with Dan Lippman at NYU and recently moved to UC San Diego to start my own group. And you'll be hearing some of the work that I have done as a postdoc fellow in Dan's lab. So we have been really interested. You have heard a little bit about the immune system this morning, looking at various cell types that's important in both health and disease. And today I want you to focus on this particular T helper 17 population. So what dictates a T helper 17 population is their ability to make very special pro-inflammatory cytokines. And this includes interleukin 17 members, and therefore you have the name as T helper 17 cells. So it seems that these T helper 17 cells are very important to fight against antimicrobial infection in the host, as well as participate in a variety of autoimmune pathologies, ranging from multiple sclerosis to cancer to IBD to arthritis. So what the lab has preliminary interested in is looking at how these T cells develop and what regulates their function. So these T cells comes in sort of variety of flavors. They started as naive T cells that start to develop in the draining lymph node, where upon antigen presentation from antigen-presenting cells, they are able to develop into their special flavors here, T helper 17, dictated by specific transcription factors such as STAT3 and RR gamma. And interestingly, these poised T helper 17 cells are particularly important in the mucosal site. So here, what we would mainly focus on is looking at the gastrointestinal tract, where epithelial layers form barrier functions to help protect the host against a variety of microbes, including commensal bacteria as well as pathogenic bacteria. And here, that's where the poised T helper 17 cells come back to these local sites of immunological challenges and decide that they can now need to convert into an effector state where they produce their effector cytokines, including those that are pro-inflammatory, such as IL-17. And we know many of these inflammatory cytokines are very important to maintain this barrier function, help epithelial cells decide that now they need to proliferate more, maintain the barrier, as well as to produce a variety of antimicrobial peptides to fight against the microbe challenges. And here are what the data actually looks like if you take a flow cytometry plot looking at the variety of T cells infiltrated into the gut. Here now you're looking at mouse, small intestine spanning across the duodenum, jejunum, and the ileum, where a variety of microbes are colonized. And here our lab has particularly interested in one commensal bacteria, the segmented filamentous bacteria, SFB for short, which is actually first described in Dan's lab with previous postdoctoral fellow Ivo Ivanov, now at Columbia. Here you can see an EM structure of these SFB bacteria colonizing directly attached to an epithelial cell at the ileum section of the mouse gut. So what you can see is for T cells that infiltrated into the gut, they actually come in, as I mentioned, a variety of different flavors. For those that goes into the ileum section of the gut, where they're now seeing this SFB-specific bacteria and antigen infiltrated, they convert it into an effector state. Here in the flow cytometry plot, you can see all the cells, all the T cells that are transcription factor RR gamma positive are now actively producing cytokines such as IL-17. However, those T helper 17 cells that protrude in other area of the gut where you do not see the active colonization of a commensal bacteria, those stay pretty much poised in the sense that they do express the transcription factor that allows them to make the cytokine, but they do not make those cytokines there. So with this observation, we became really interested to try to understand what makes a particular segment of the GI tract sort of allowing signals to activate or convert these RR gamma positive T helper 17 cells into an effector state. And it turns out some of these molecules that are produced locally by these epithelial cells that are seeing the commensal bacterial colonization include one of the molecules, which is named here, serum amyloid A, or SAA for short. SAA1 and SAA2 exist in a cluster on the genome, and they are actually both highly induced upon the colonization of this SFB bacteria. So here now you're looking at total tissue extraction from the small intestine, the duodenum, or the ileum portion of the small bowel from mice that are colonized either with or without SFB. So you can see commensal colonized ileum section of the gut are inducing a very high abundance of this SAA molecule. So what is an SAA molecule? It's actually a very acute response protein that are primarily found expressed in the liver during acute injury. Actually, very interesting in its ability to associate and couple with HDLs in the serum. And pathologically, it's been associated with a variety of autoimmune disease. So if you look at patient joints that are inflamed, patients with arthritis, they have an increased level of SAA. Also, if you look at patients with MS, multiple sclerosis, they also have an elevated level of serum amyloid A. So the real question is, what is this serum amyloid A doing that is locally produced by the ileum epithelial cells during bacterial colonization? So with this experiment, what we did is genetically delete SAA1 and 2 in the genome cluster and ask what happened to these animals when we colonize with bacteria. Again, this is a commensal bacteria of SFB, which we can introduce by oral gavage. So what you can see is in the different segment of the small intestine, the duodenum or the ileum, the percentage of RR gamma positive poised T helper 17 cells are pretty similar between wild type and double knockout animals. However, if you look at the ability of these these poised T helper 17 cells to become effector, meaning that their ability to produce now this inflammatory cytokine is actually very significantly reduced in the SA1 to double knockout animal. So the real question is, what is this external cue coming from the epithelial cells? How does it signal to T cells? What are some of the downstream recognition factors in, for example, the nucleus that recognize this environmental cue and drive this effector conversion? So what we can do is mimic what we are seeing in vivo using an in vitro system where you can sort out naive T cells, culture them in vitro, now using T cell receptor activation mimics with antibody against CD3 and CD28, along with cocktails that drive them towards the T helper 17 lineage. And now in the presence or absence of SAA, mimicking the environment from the ileum, or let's say duodenum section of the gut, what are you seeing? How is the T cells reacting to the environmental change? And one of the top motifs, it turns out when we done RNA-seq experiment looking at these cell lysates, cultured T helper 17 lysate, is a transcription factor motif for ROR. So as I mentioned briefly, RR gamma is the key master regulator of T helper 17 lineage. And here, using a luciferase reporter assay, we confirm that in cultured T helper 17 cells, cultured in the presence of SAA, an RR gamma reporter is actually significantly induced, suggesting that this external cued molecule coming from epithelial cells is indeed acting on a transcriptional machinery in T cells. So one of the things we have become really interested in is this transcription factor RI gamma. So in the next part of the story, we want to briefly tell you a little bit, another story that we have looked at is how RI gamma, which is a nuclear hormone family receptor, actually contribute to T helper 17 biology. So we know that nuclear receptors, such as those of RI gamma, interact with a variety of co-activators and co-repressors, in addition to what we already just talked about, RNA polymerase II, to drive RNA transcription. So we became really interested to try to understand what are some of these missing black boxes, the co-activators and co-repressors that needs to drive T helper 17 activity in vivo. So with this system, we were able to use our primary culture of T helper 17 cells, perform a mass spec assay using immunoprecipitate materials against RI gamma. So with this, essentially, we identify close to 20 very highly confident hits that are suggested of co-regulators for this transcription factor in Th17 cells, which we follow. Right now, I just want to give you a summary of what we found. Essentially, we found one of the RNA helicase family, DDX5, to be a co-activator of RI gamma, work together with a non-coding RNA RMRP. So what is DDX5? So DDX5, as I mentioned, is an RNA helicase, meaning that it can utilize ATP, perform ATP hydrolysis, and use that energy to modulate RNA secondary structure. So DDX5 in particular is a member of the dead box family, and actually it has been previously described to have roles in transcription as well as a ribosome biogenesis. So we began to really try to understand what exactly is the role of DDX5 in T cells. Using a T cell conditional knockout animal that we developed in collaboration with Francis Fuller-Pace from University of Dundee, we were able to go back to our mouse model. Again, you are looking at animals that are breed in a bacteria-negative condition, which then we reintroduce this commensal SFB bacteria. And then here you can look in vivo in the gut of these animals and ask what happened to their T cells. Are they able to develop? Are they able to commit to the Th17 lineage, express the proper transcription factor? And here it shows that indeed it can. However, if you look at the cells that became committed and ask what's their ability to make effector cytokines, you can see DDX5 deficient animals, specifically in T cells, lack the ability to make effector molecules. So this is in steady state. What about disease? I mentioned to you these T helper 17 cells are very important in both containing both commensal as well as pathogenic bacteria in, for example, mucosal sites such as the gut. Also very much important in driving autoimmune pathologies. So one of the models we have been using is looking at a colitis model. So this is a T cell-driven colitis model in mice, where you can introduce naive T cells to RAC recipients, and those recipients that are receiving wild-type T cells will eventually develop chronic colitis in the span of 8 weeks. However, if you introduce, instead of wild-type T cells, an RI gamma, so this transcription factor deficient T cell, now these recipient animals do not get sick, so they are relatively protected from an autoimmune colitis. So the question here is, for the molecules we identify, such as DDX5, if indeed they work together with RI gamma, can we test this idea in mouse model now? So what we went back to do is basically take out wild-type or DDX5 deficient T cells, now introduce them into a variety of RAC recipients, and ask how does this recipients react in terms of chronic colitis development. So you can see those that receive wild-type here in red are dropping in weight. However, those animals receiving DDX5 deficient T cells are relatively staying in normal weight scale compared to the PBS control. And also, we have looked at the colon of these animals. Again, wild-type recipients get very severe inflammation. You can see by lots of neutrophil infiltration and destruction of tissue morphology. And those animals receiving the DDX5 deficient T cells do not develop colitis. So with that, we became really interested in understanding how DDX5 as an RNA binding helicase in fact contributes to its role in Th17 biology. So what we were able to do is go back to do a very simple assay, take naive T cells from these DDX5 deficient T cells and reintroduce either wild-type DDX5 or an RNA helicase null mutant of DDX5 and ask whether the activity of DDX5 requires helicase activity. And here you can see on this result panel that if you reintroduce DDX5 wild type onto the deficient background, cytokine production is completely rescued. However, if you look at cells that are receiving the RNA helicase dead mutant of DDX5, its ability to make cytokine remain inefficient. So with that, we became really interested to identify the actual RNA substrate of DDX5's RNA helicase activity. For that, we took advantage of the RIP-seq technology, where we use antibodies specifically against either DDX5 or RI gamma, its interacting partner, and then pull down these proteins respectively, perform deep sequencing to understand what are some of the RNA species that are interacting with the protein complex. And here is showing you what our top candidates look like, highly enriched 20 non-coding RNAs in Th17 cells. And one of these non-coding RNA is very highly enriched in both DDX5 as well as the RI gamma immunoprecipitation. And turns out RMRP, which is the one that we next focused on, is actually a very evolutionarily conserved non-coding RNA. It's actually one of the first of its class to be non-coding RNA and associated with a human disease. So the human disease is called cartilage hair hypoplasia. So these patients have a variety of syndromes going from skeletal dysplasia, hypoplastic hair, as well as neuronal dysplasia in the intestine. And from our perspective, even more intriguingly, is that these patients have defective immune system, particularly in the T cell compartment. Many of these patients do survive and do grow into adulthood and have relatively normal life, with the exception of many that suffer from recurrent infection and autoimmune pathologies. And it's been difficult to model this human disease in animals, mainly because other groups have tried to generate an RMRP knockout animal, and it turned out to be embryonic lethal. So we have also started collaborating with Joanne Walter at MGH and many other physicians at Mayo Clinic and Clinic for Special Children. And there we were able to collect a group of CHH patients, approximately 10 of them, to look at their ability of T cells from these healthy and CHH controls and their ability to make cytokines or produce T helper 17 cells. So you can see CHH patient T cells indeed seem to lack or have much reduced ability to make inflammatory cytokines such as IL-17F. And as I mentioned, this RMRP is a long non-coding RNA that's very atypical in the sense it's very well conserved between mouse and human. And here you can see the alignment between human RMRP and mouse RMRP, and highlighted as some of the CHH patient mutation that are actually conserved in mouse. So in the lab, we have become interested in adopting the CRISPR-Cas technology to introduce human CHH patient mutation into the mouse genome and utilize the mouse genomics to tell us what's going on and how these non-coding RNA in fact function. So the last slide I'll tell you, just this briefly, this one mutation in CHH patient, G262, corresponding to G27 in mouse. What happened is that we were able to generate this total knock-in animal at the endogenous RMRP locus. And now if you take out naive T cells from these donor animals and put them into the RAC recipient in the T cell-mediated colitis model, you can see that similar to what we observed for DDX5 as well as RI gamma deficiency, mutation in this single residue on this non-coding RNA also conferred T cells inability to induce colitis in this T cell mediated model. So with that, I just want to finish with one last slide where you can look at the location of RMRP by RNA fish. It seems that this non-coding RNA localized in a very specific compartment in the genome, in the nucleus, using ChIRP-seq technology that's initially developed by Howard Chang and others. We were able to localize the location of this non-coding RNA in the mouse Th17 cell genome. It seems to be very highly enriched, where this transcription factor RR gamma is also occupied. And interestingly, its genomic occupation really depends on this RNA binding protein DDX5. So typically in wild-type Th17 cells, you can recover approximately 2,000 peaks enriched with this non-coding RNA RMRP. In Th17 cells in the DDX5 deficient background, you lose essentially 2/3 of the non-coding RNA binding sites. So with that, I would like to close and really thank my postdoctoral work supervisor, Dan Littman, and many wonderful colleagues from Dan's lab, and many collaborators outside of NYU from Howard Chang's lab, as well as Francis Fuller, Pace Dundee, and Frank Rugo, who helped us a lot also with antisense oligonucleotides. And with that, I would like to thank you for your attention, and I'll take questions.